Now, just a quick disclaimer. Graduate is a debated question of my circumstance. I was a student here for so long, I think they lost the records. And I must say, to see so many of my old mentors and classmates online and colleagues, it's great to have you here to listen to my talk. So this is really a story about One Health and how this multidiscipline really has been part of the path that I've accidentally come across during the course of my career. And I just wanted to make a comment. When we started, or at least tried in Cambridge, to initiate a One Health program about 10 years ago, we went to our medical colleagues really enthusiastically, thinking they'd embraced this. And we said, well, we'd really appreciate your involvement. And they said, One Health? What do you mean? It's our health. And they just didn't get the interdisciplinary aspect of it, and they were more interested in animal models of diseases, human diseases. Suffice to say that paradigm has shifted significantly. Today, what I hope to touch upon is what led us to some new technology. Hopefully, that'll help us prevent future pandemics by making vaccines that have very broad protection against some of those notorious viruses that threaten us and that are highly contagious. So I'm going to focus on zoonotic diseases. RNA viruses are the most notorious of those. They have variable genomes, and they, because they change, they adapt to other species when they come in contact with them. And I'll take you through some examples. I'll take you also through a real-life example of an outbreak in Africa and lessons that we've learned from that, really the largest global outbreak of Ebola that mankind has faced. And then the information that we gained from that experience was that we could use the sequence from these pathogens actually not only to track and trace or contact trace individuals who've been infected and passed on the virus to others, but it allowed us to understand what regions of these viruses remained constant and what changed and what they were doing to escape our immune responses. So the second is going to be the innovation, the technical solution we have to try to get around this hypervariable problem of these RNA viruses. And I'll give you some example of what we called, or what was recently described as digitally immune optimized synthetic vaccines. And I'll use that acronym DVX for short. And then I'll talk probably about one of the most topical challenges and current threats we have, and that is the threat of an influenza pandemic. And I'll give you examples of past human pandemics of influenza and how H5 is trying very hard to set a new paradigm. Just to give you some background, because I'm going to talk about R0 in a couple of slides, but this individual here in the middle is us. And we all get exposed to viruses throughout the course of our lifetime. And those viruses imprint an immune response. We have B cells and T cells for our adaptive immune responses. It recognizes different parts of these viruses. But actually, we could probably tell which decade everybody here in the audience has been born in by the type of influenzas that they have the strongest response to. And that's because the B cells are imprinted on that first virus. That's a great thing. It has saved us, and we can boost it with current vaccines. But it's also a problem because it helps or prevents us from shifting to new strains, or at least really refocusing those immune responses on new strains. And of course, the current vaccine technology up until now have been to take virus isolates that have been well characterized by global institutions such as the WHO, and to select those for vaccine strains. And every year we're offered, once you reach 65, you're offered an influenza vaccination, and that contains these wild type isolates that are predominant and expected to become dominant the next season. However, they only pick one isolate, and don't forget, they're picking it from something that's already escaped our immune responses. So in many ways, it's a problem. So those pre-exposure histories that we all have and that differ amongst everybody in this room influence the way we will respond. Maybe why some people will catch a particular cold, although the same person sneezed on them. One doesn't get infected and one gets really ill. And of course, just put that in the context of the population of animals out there that have their own ecosystem of viruses and pathogens. And because of domestic practices, you know, of all in, all out, you have naive populations who are fantastic reservoirs for a new outbreak unless they're properly immunized. And of course, we've seen a remarkable feat of viral adaptation with H5N1 in dairy cows. And this, I'm talking about the clade 2.3.44b that has managed to use the sialic acids from airways to really adapt well to the mammary gland of cattle. And of course, in America, before they had stringent controls, you could go and buy raw milk and isolate or even get PCR positive milk from your local grocery store. So it was a big problem because it's now in the human food chain in many respects. I'll talk about some of the pathogens in Africa that are current and endemic and some of the surprises that come along like Ebola. So this is just an old diagram from Diamond and Wolf about really the adaptations that various viruses go as they climb the species ladder, if you like, to humans. Rabies, of course, we're all familiar with. Many of us veterinarians have titers, so we're hopefully protected, but it's of high consequence, even though it has a low R0, because it is often fatal. Ebola, on the other hand, has a natural reservoir in bats or in bushmeat, and of course, spillovers from human to human occur, but if enough humans are infected and you have a local outbreak, well, the consequences are quite dramatic, and we'll talk about that in a minute. And then, of course, there are these stealth outbreaks that have become pandemic. The HIV virus evolved from primates, and in fact, it was chimpanzees who could hunt different kinds of monkeys that, well, I guess one very prolific hunter managed to eat in a particular season a couple of monkeys with different parts of a genome that actually came together that formed the ancestral HIV strain. And of course, the R0 of HIV in a naive population can get up to 1.9 in a particular instance. And again, it depends on immunity. So here we're talking about naive populations. And these go pandemic when that R0 is above 1 or 2. And in fact, COVID started out at about 3.4. The different variants in different populations then had higher R0s. So let's talk about what happened in West Africa in 2013. So Guinea, in the place, in the initial outbreak, a place called Geketu, close to Miliandu, the index village, it had the first cases, but they didn't know it was Ebola. And that's because they didn't have the diagnostic reagents, nor did they even expect that it would Ebola, because it didn't occur in that part of Africa. And there was a delay in the diagnosis of that, and a delay in the international response, hence, because they presumed it was the endemic hemorrhagic fever caused by Lassa virus. Cholera was another, but it wasn't only until they sent samples actually to Europe for diagnostic testing that they identified Ebola. And then, of course, the response was quite dramatic. So this is a picture I took from a Jeep going into an Ebola quarantine center. And I'm going to show you a few snapshots and the consequences of what we learned. This was a team that we were traveling around Guinea with. Guinea is a mosaic of different ethnic cultures and languages, and so we had a remarkable linguist here, Raymond, second from the left. Farthest from the left is Dr. Louisa, who is an infectious disease specialist in Lassa fever. In the middle is a veterinarian, again, a linguist who spoke many of the languages. And to his left, our right, is a wildlife expert. And we went looking for how this virus got from Central or East Africa all the way over to West Africa. And that was one of our missions. The other mission was to understand what was going on with Ebola survivors. Now here is the geographic map overlaid with different countries that have had one or more different hemorrhagic fever outbreak. These are all the countries that have had Lassa or a combination of Ebola, Marburg, or the various Ebola-related viruses such as Bundibugyo or Ebola Sudan, etc. And on the right is the same area of Africa that has really outbreaks of different clades or lineages of Mpox. So we figured that if diagnostics on the ground were delaying the identification of outbreaks in order to find the right potential vaccine or preventative measure, that we needed to have a broad-based vaccine, a vaccine that could be used in all throughout sub-Saharan Africa to cover these diseases. Now, how could we do that? How could we achieve that? Now, it was really the study of individuals who are in the background of this tent here, the Ebola survivors. And at that time, survivorship was improving because of the heroics of clinical care. But there was a concern that those survivors needed a home to go back to. There was just so many of Ebola survivors, they couldn't keep them in these quarantine facilities. So there was a great movement to be able to triage them so they could be rehabilitated and go home to their villages. So two negative PCRs, were the main criteria and symptom-free before they were released and allowed to go home. However, later they discovered that the virus was still at least detectable. In fact, one American physician who had really almost died of Ebola subjected himself to really intensive investigation. They even took aqueous humor from his eye, never mind cerebral spinal fluid and all sorts of other places. And they even found by PCR remnants of the Ebola virus genome. So it suggested to me this virus could persist. So it was a real conundrum. How do you then advise these survivors and the villagers to accept them back home? And this was really the big debate at the WHO meeting that we had in Freetown, Sierra Leone. So this is the paradigm that there are reservoirs, such as bats that carry these filoviruses, and that these bats, fruit-eating bats, often compete with other mammals that like to eat fruit, often under the same tree. And of course, these monkeys or bushmeat hosts that do get infected, of course, are often hunted and taken home for meals. And that's what we suspect may have happened in Miliandu. So I mentioned that you can get an individual who gets infected, is well treated, and who survives, but may still potentially have that virus reservoir in their body. So this is a paper we published on this hypothesis and this concern that humans could also be a reservoir for this virus. And that played out actually several years later. This paper was in 2015. The finding of Ebola in an Ebola survivor in Guinea was published in 2021. So some years later, this woman with her new husband had a baby, and of course during that cesarean to rescue the baby, members of the family also became ill and some of them perished. And this all was evident. Why? because we could look at the genome that was associated with the first outbreak here. This is a sample from different individuals in 2014, if you can see this laser printer. And here's that isolate from that new outbreak in 2021. And there is so little difference by the branch length, okay? The horizontal branch length shows the virtual similarities between these viruses. Okay, so how could this use of genomics then help us in the future combat these potential outbreaks? And I'm showing you some of the clues here. Depending where you look in the genome of different viruses, you can look for similarities. Places in the genome or protein structures that actually can't be changed through evolutionary adaptation. And this is, again, another picture I took inside an Ebola treatment camp. And here we're using a little MinION sequencer to get that particular sequence from a particular individual. And that allowed not only, as I mentioned, track and tracing, but it gave us that essential information. So we came back to Cambridge and got together with some experts and came up with this process where we took viral genomes and developed a vaccine pipeline. And that pipeline was to take these sequences from outbreaks and looked at the parts of these viruses that couldn't or didn't change and focused on understanding which immune responses actually targeted these types of proteins. And so, as I mentioned, we called it digital immune optimized synthetic vaccines. This is Simon Frost, who's a computational biologist who did that early machine learning. And this is Ralph Wagner, who developed a company making synthetic genes. And many researchers will be familiar with GeneArt and may have ordered gene sequences from GeneArt. And this is Rebecca Kinsley, who's now the CEO of Diosynvax, who actually had a pipeline of neutralization assays to be able to look whether or not we were getting broad immune responses with candidate vaccines after we vaccinated mice. And that technology worked, and it was a spin out of the university because it needed investment in order to make potential vaccines for events that had yet to happen in the future. Our first candidate was one for hemorrhagic fevers. And because the successful use of attenuated smallpox vaccines are also useful for preventing mpox infection, we decided to use those same vaccines from smallpox. And this is one called MVA, Modified Vaccinia Ankara, that was used in the smallpox eradication program in Turkey. And we put the different genes that we had identified from our analysis to be expressed in this mpox vector. And here's an example of those different structures. And I'm going to just go over this very quickly, but suffice to say, we could get good immunological breadth despite the high variability of these viruses that we are trying to combat. So we then took this virus candidate to Marburg. Also is another big problem because Marburg also was previously restricted to the Rift Valley area, in particular caves that bats roosted in, but now it's found on both coasts of Africa. So these bats, through whatever reasons, climate change or dynamics in the changes of migration, that disease now is on both coasts and affects many different countries. So we took this vaccine to Winnipeg, to the National Microbiology Lab, and they had in their freezers Ebola Sudan, Marburg virus, and Lassa fever, three very, very different viruses. And I'm showing you here the survival curves of the vaccinees, the ones that received this vaccine, here in color, and the black represent the controls. And you can see the days of survival. And this is showing you that the vaccine is working 100% in the case of Ebola Sudan, 100% in the case of Marburg, and the same with regard to Lassa. And I'm happy to share the reference for this paper after the talk if anyone's interested. So suffice to say, we've been fairly rigorous in our analysis and showing protection. And we're now trying to get the world to think about manufacturing these kind of vaccines to prevent these future spillovers in areas where they are endemic. Now, many of us, of course, remember COVID dramatically. Same problem as I described earlier. The first isolate that was used in the first vaccines was an isolate from Wuhan. And that Wuhan strain went into the first vaccines. Again, the strain that had actually spilled over into humans. And we know that this group of coronaviruses, these beta coronaviruses, have caused actually 3 spillovers into humans. One, those you may remember, in 2003, the SARS outbreak that came following a wedding in Hong Kong and infected families and contacts in Vancouver and Toronto, as well as different cities in the States. But it was because this virus had a lower R0 than since SARS-CoV-2, the current COVID, they were able to contain it and actually detect people who had fever by using infrared thermometry to then quarantine individuals. And quarantine worked in the case of SARS-1, if you like. So we decided that this was a high risk, that there could be more spillovers from this group of coronaviruses. Let's see if we can do the same thing and make a vaccine that would protect from all of these. And this is Sneha Vishwanath, who did really the computational work and developed this structure, which is I wouldn't say a chimeric protein. It represents different epitopes on the structure of this protein of a sarbecovirus. And the sarbecoviruses are those large group of viruses that are common amongst different bat species, especially in Southeast Asia, that can spill over to humans. And they have spilled over to humans. Instead of waiting for the next pandemic, why don't we try to get a vaccine ready so that it can be launched immediately and get something that's broader, so we're just not using one strain and getting into that same problem of deja vu. This is the selection of that particular one candidate in that box there that eventually was taken into a human trial. So this vaccine then was put into one, and it was challenged again by our colleagues at the NML with the original SARS virus. This happens to be an isolate from Toronto. MERS, which spills over from camel racers, camels, and in fact it's found in camels throughout North Africa, but yet to have clinical cases from North Africa, but the Asian Peninsula, and then SARS-CoV-2. And again, the vaccine candidates are those ones in red or blue, and they are actually doing what they were designed to do. They were protecting not only from weight loss and fever, but keeping those animals alive without weight loss. So let's just get back to the problem of using wild-type isolates. So everybody jumped on the bandwagon and used that isolate from Wuhan, or the sequence of that spike protein, or the RBD, that was made available in mid-January 2020. But we know that these different variants started to escape these vaccine responses, and every year subsequently, there were boosts that were offered. But these boosts consisted of isolates that were chosen that were potentially themselves escape variants. This just tells you that the current practice of taking isolates and using them as vaccines have significant consequences. The current flu vaccine, although I would recommend everybody who is at risk get it, and I have, these still are only, on a bad year, 29% efficacious, and in a good year, only 58%. Not very, very good. but it's really all the industry has right now. And this, of course, is the problem. The problem is that you take the isolate, and it takes not only time to characterize it and develop all the criteria for the vaccine, but by the time the vaccine is manufactured, another variant has come along and is circulating in the human population. I'm going to skip over this. It's a bit of detailed modeling that actually shows the number of lives saved if such a broadly protective vaccine was available at the day or the week of a potential outbreak. And we've sort of categorized these if it was available for immediate use. And you can see the impact on the number of lives saved here. Or phase 3 ready. So that means it would go into a clinical trial where people were affected and could be tested. Okay, I want to get to influenza. Influenza, in contrast to these other examples, has a unique genome. It has 8 segments. And as I said earlier, if 2 or more influenza A viruses infect the same individual animal, it's like a deck of cards. If they infect the same cell, those genes can reassort. And that's what we know in the field as reassortant viruses. And it's expected that that is one of the major mechanisms of how we get what's called antigenic shift, a profound change in the immunogenicity of that particular variant. Now, the other problem is, of course, influenza A viruses infect many different species. And that's what we're seeing not only now with the bird flu epizoonotic, and the cases that we've seen not only in dairy cattle, but actually in this particular sheep in Yorkshire, but also in pigs. And this is a problem for veterinarians because these pigs are a reservoir of important potential human pandemic virus. In 2009, we had a swine flu pandemic. Many of you may recall that. And that swine flu was a reassortant from swine to human. And what we have right now is a complex mosaic, and I'll show that in subsequent slides, of different influenza A viruses in birds that are reassorting and causing significant deaths and fatalities, not only in waterfowl, but also in certain mammal species. So this isn't too far from where I live in Cambridge. And I was driving by this pig farm one Sunday morning, and I thought, I've got to get this in action. Because here, we've got these migratory birds flying over free-ranging pigs. And of course, everybody wants free-ranging pigs because they're not stressed. And of course, the chickens are allowed to stay indoors, but the pigs aren't. And hence, we're just waiting for a natural accident to happen. I mentioned that there's a good, solid history of these reassortants spilling over and causing pandemics in humans. And of course, the first significant example of that is the 1918 Spanish flu. It killed more than 20 million people. Of course, then we had H2N2. That is an avian type influenza, and those who were born before 1957 may have an immune response to that pandemic virus. Subsequently, we have been exposed to it, of course, and those individuals of us who experienced that new pandemic virus will have imprinted that particular response. Hong Kong influenza, here's an H3N2 introduction, and again, subsequently, as I mentioned just earlier, that pandemic swine flu from 2009. And here, clearly the animals that have been involved in that evolution of those human pathogens shown here pictorially. So again, we've used this process to come up with vaccines that could give that broad protection. And we first applied this as sort of a research project to look at swine flus and ask, could we make a virus that would protect against those swines that are zoonotic from pigs that have jumped to humans versus potentially pandemic, and I've got these listed over here. And of course, that could also protect against the circulating human H1N1 viruses. And we're showing here in red here the immune responses induced by this digital vaccine versus a wild type WHO strain, H1, recommended for the seasonal flu usage. And you can see the breadth that this responses give from this particular digital vaccine right across the spectrum of different swine pre-pandemic and pandemic, including the 1918 influenza strain. So we were quite pleased that we could get that breadth of protection in terms of neutralization and protection from infected cells. We then went to the local APHA, the Animal, Plant and Health Protection Agency in England, who had this fantastic swine model where pigs could be put into high containment and challenged with different influenza strains. And so we thought we'd do an exercise using their respiratory model where pigs would be exposed to virus intranasally. And here I'm showing you in black here, naive pigs that are exposed to a particular strain. We'll call it A swine England. Okay, this is the ones in black, and the black ones have not been vaccinated. And each of these animals is developing infectious virus in their respiratory tract, in their oropharynx. And we're also detecting T cells responses, so that means that the virus is replicating and the immune responses is responding to that virus before it's cleared, okay? It's a non-lethal infection in swine. So if you take then that strain, same strain that has caused those symptoms in that pig or in those pigs and infect them with that same strain or vaccinate them with that same strain, they're protected. And that is what's going on here with this green line here. So the animals that have been challenged with this same virus, if they've received the same virus but inactivated, they're protected, or at least the threshold of replicating viruses is severely blunted, hopefully sufficient to protect or prevent onward transmission. What is interesting here is that if you take a virus strain that is somewhat different, A. Victoria, for instance, and challenge those animals that have been vaccinated with a different vaccine than the challenge strain, you'll see that there's absolutely no difference than just naive animals who've been infected. So just simply by taking a different strain of virus, this was 2019 versus 2009, no protection. And then if you take this digital vaccine, the same one that I showed you that gets that broad protection, you're able to get the same effect as if it was a strain-based, strain-matched vaccine. Okay, so the complexity of H5NX, as you know, many of you have heard about H5N1 and the spillovers and the human cases, but certainly the bovine cases, dairy cow cases in North America. But we hear very little about what's going on in Southeast Asia, where they have a lot of different mixed animal markets, where pigs and birds and humans all come together, and live animals are exchanged and sold and slaughtered. And in fact, what's happening there is double digits of infections in the previous year of human cases and some significant fatalities. So this is the family tree of the H5N1 or NX viruses. And it starts here with mongoose guangdong, and of course over time you'll see that little branches have broken off from this radial tree, and each one of them have been given a designation a clade designation by the WHO. And the ones that are causing human cases in Southeast Asia are a very different clade called 2.3.1c versus the one that's causing problems right now in North America. And I just wanted to show you this slide. And this is just looking at one protein, the hemagglutinin protein, which is very important for virus entry, and the amount of variation over time. Now, of course, there's a bias because of sample selection. But as you can see, as we go from the 60s here to the 2000s and beyond, the virus has dramatically expanded. And with that, the number of animals that other species that have become infected. And what I've indicated here are the numbers of different types of neuraminidase that the virus is reassorted with. So neuraminidase is on a different gene segment than hemagglutinin. And you can see as this diversification increases, so have the different partners of neuraminidase. And why is this important? It's because that we, as humans, have immunity to some of these neuraminases, but not others. And it's that naive population that I alluded to where you can get that very high zero in human-to-human transmission. Now, there's other things, fortunately, that the virus needs to have or change to be able to adapt efficiently to humans. But it's with this information we can build better vaccines. And this is just showing you, again, one of those digital candidates, and the significant breadth in this heat map that I'm showing you against all these different clades from that radial tree. So we have an exciting candidate that we've put together based on these. So it gives very broad neutralization, better than these strain-based vaccines, which I'm showing you over here on the right-hand side. So again, these red immunized animals are getting broad hemagglutinin and neuraminidase neutralization. So again, we took these vaccines and sent them to Winnipeg, where they did some parallel challenges, and I mean parallel challenges. So they had one group where they had an old isolate, here this clade 2.3.21c, this is clade 1, this is Vietnam, and the current circulating North American problem, 2.3.4b. And this was from the BC, British Columbian patient who became very ill more than a year ago. Now, here we're showing in red on this diagram on the far right, you can see what I've shown, this red line that encompasses all these different clades. That's the digitally immune-optimized vaccine, and we're challenging either, or vaccinating either with it, and challenging either with this clade here, this clade, or this clade. And these are all color-coded. And you can see that the breadth of protection is related to really all of those different potentially circulating clades. And that represents over 20 years of virus evolution. The comparison are the WHO inactivated viruses that are currently used and stockpiled for H5 vaccines. And you can see that they're only partially protective even when they're relatively closely related. Okay, so we have a candidate that has that broad protection. It induces not only hemagglutinin and neuraminidase against not just H5N1, but H5N6 and other neuraminases that are causing problems in different parts of the world. So a single vaccine is available if we can motivate governments to stockpile such a vaccine. And this kind of technology is currently being used by the company to make universal flu vaccines and better human seasonal flu vaccines. And as I've shown you, the efficacy that we've achieved is as broad as 20 years in some cases in the cases of the human seasonals, we've been able to achieve up to 5 years of protection with the H1N1 viruses. And we've been able to deliver this vaccine by a large number of different modalities, not just RNA, but protein, recombinant viral vectors, and nanoparticles. And they've all seemed to be equally as effective. So I'd like to stop there and take any questions that you may have. Thank you.